Hello, this is Jeremy James, plant physiologist from the Eastern Oregon Agricultural Research Center, here to present an Ag Minute. This week we'll be discussing how stable isotopes can be used to better understand how rangeland ecosystems function. Right now I bet you're thinking, whoa, hold up, did he just say stable isotope? As the distant memories of science class begin to take shape. Well folks, you heard right. Stable isotopes are actually a very powerful tool that we are using in some research at the station. For example, we can use stable isotopes to measure water stress in plants, as well as water and nitrogen flow through ecosystems. While the topic of stable isotopes might seem a bit technical for this particular portion of your day, the basic concept of how we use these tools is fairly simple, and we'll break it down to its basic steps this week. An isotope is simply an atom of an element that contains a different number of neutrons than the other atoms of the same element. The key point here is that these neutrons, which are just particles of the atom that have no net electrical charge, are pretty darn heavy. This means that different isotopes of the same element will have different weights. This simple difference in weights is one very important attribute that allows isotopes to be so useful. A second important attribute is that the different isotopes of the same element differ a lot in their abundance in nature. For example, 99.9% of the hydrogen in the Earth is in the form of hydrogen 1. About 0.01% of the hydrogen in the Earth, which we call hydrogen 2, has an extra neutron and is a lot heavier than hydrogen 1. The third nice attribute of stable isotopes is they are not radioactive and are therefore safe to use. This week we are discussing how stable isotopes can be used to better understand how rangeland ecosystems function. Isotopes are simply atoms of the same element that have different weights. There is usually one isotope that is common in nature and one isotope of the same element that is very rare. A lot of rangeland research focuses on how the elements, carbon, hydrogen, and nitrogen, flow through the system, and these three elements all have at least one stable isotope. Carbon isotopes can tell us a lot about the production and photosynthesis of plants. Since water is H2O, hydrogen isotopes can tell us a lot about water flow through the system, and nitrogen isotopes can tell us a lot about soil fertility. Before discussing how we use these isotopes in research, let's explore how isotopes can be measured. As we mentioned, different isotopes of the same element will have different weights. A mass spectrometer uses this difference in weight to measure how much isotope is in a sample. Here's how it works. The sample is combusted to a gas and broken down into charged particles. These particles are sent through a chamber into a magnetic field. As the charged particles move through the magnetic field, they bend. The amount a particle bends, however, will depend on its weight. It's like rolling a large ball bearing or a BB past a magnet. A small BB rolled past a magnet would have a greater tendency to bend toward the magnet than the ball bearing. A detector measures how far each atom bends and can therefore tell us how much isotope is in a sample. This week we are discussing how stable isotopes can be used to better understand how rangeland ecosystems function. Remember, isotopes are simply atoms of the same element that have different weights. Carbon has two stable isotopes. About 98.9% of the carbon on the Earth is in the form of the lighter carbon isotope 12C, and only about 1% of the carbon is in the form of the heavy carbon isotope 13C. Even though the heavy 13C isotope is not very abundant, the amount of 13C in a plant can tell us a lot about how well a plant is growing in a particular environment. Here's how this works. The carbon dioxide in the air diffuses into leaf cells through open pores on the leaf surface. Once inside the leaf, enzymes convert carbon dioxide to food for the plant. The enzymes that do this, however, favor the lighter 12C isotope over the heavier 13C isotope. This means that when the pores are open and plenty of carbon dioxide is diffusing into the leaf, there will not be much 13C incorporated into the plant tissue. When water is scarce, however, plants close the pores on the leaf surface, so water cannot escape from the plant. This means that very little carbon dioxide will diffuse into the leaf cell. As a result, the enzymes that convert carbon dioxide to food will not have the luxury of choosing the lighter 12C isotope over the heavier 13C isotope. Because plants that are under water stress will have more 13C in the plant tissue than plants not under water stress, 13C isotope data can be a good indicator of plant water stress. These 13C data also can be used in crop breeding programs to screen for drought-tolerant plant varieties. This week we are discussing how stable isotopes can be used to better understand how rangeland ecosystems function. Stable isotopes are simply atoms of the same element that have different weights. Hydrogen has two stable isotopes. about 99.9% of the hydrogen on the planet is in the form of the lighter hydrogen isotope 1H, and only about 0.01% of the hydrogen is in the form of the heavy hydrogen isotope 2H. Because the heavy 2H isotope is not very abundant, scientists can add small amounts of water that has been enriched with 2H to trace water movement and use by plants. Here's how this works. In the lab, scientists add a lot of 2H to regular water that is usually made up of 1H. The water, now labeled with 2H, is applied to experimental plots as a tracer to look at water flow through the soil and plant. Using this approach, researchers have shown that different rangeland species often take up water from different locations in the soil profile. For example, bunch grasses usually take up a lot of water from the upper soil layers, while shrubs and trees often take up water from deeper soil layers. Using water enriched with a heavy hydrogen isotope, researchers also have shown that the soil depth that certain species can capture water can shift depending on what other species the plant is growing with. For example, sagebrush can be restricted in using deep soil water when surrounded by juniper. Researchers have also shown, with water enriched with a heavy hydrogen isotope, that some species can actually take advantage of small amounts of precipitation that arrive with summer thunderstorms, while other species cannot. This week we are discussing how stable isotopes can be used to better understand how rangeland ecosystems function. Stable isotopes are simply atoms of the same element that have different weights. Rangeland scientists know that the amount of nitrogen in the soil can be an important factor influencing the spread of weeds. Nitrogen has two stable isotopes. About 99.6% of the nitrogen on the planet is in the form of the lighter nitrogen isotope 14N, and only about 0.3% of the nitrogen is in the form of the heavy nitrogen isotope 15N. Because the heavy 15N isotope is not very abundant, scientists can add small amounts of solution enriched with 15N to trace where nitrogen flows in an ecosystem. This can allow us to determine how different management treatments influence soil nitrogen availability, which plants compete better for soil nitrogen, and how weeds might influence the availability and cycling of soil nitrogen in rangeland systems. For example, some research at the station using 15N isotopes have shown that when medusa head invades, the soil nitrogen cycling can be altered to positively affect the growth of medusa head and negatively affect the growth of native bunch grasses. This indicates that restoring areas infested with medusa head may require more than just killing the weed. We may also need to consider management treatments that can return the soil nitrogen cycle back to its natural state. These examples that we have talked about the last couple days show that stable isotopes are a pretty handy tool that has let rangeland scientists get a much better understanding about how these systems work than would otherwise be possible. Thanks for listening and have a great weekend.